Hello, everyone, and welcome to Neurocracy. Today, we're joined by Dr. Erin Gibson, who is an assistant professor of psychiatry and behavioral sciences at Stanford and a member of Stanford's Center for Sleep and Circadian Sciences. Her lab investigates how the circadian clock regulates glial cells and how this regulation impacts myelination, sleep architecture, and brain health. Dr. Gibson, thank you for joining me today.
Thanks for having me.
So, my first question is, how did you get involved or interested in sleep research?
It started back when I was in grad school. I originally had gone to grad school. I did my PhD at Berkeley, and I had gone to actually study neuroendocrine things. I was interested in sort of the brain hormone intersection. But I quickly fell in love with the role the circadian system played in regulating that process because virtually all hormones are modulated by the brain's clock, the suprachiasmatic nucleus. So we have a clock in our brain that allows us to sort of, as organisms, oscillate on a 24-hour cycle, even irrespective of the 24-hour environmental cues. And the circadian system also regulates the sleep-wake cycle. So there are two things that drive the sleep sort of need. There's a homeostatic drive, which we all have experienced if we stay up too long or too late, and we eventually can't keep our eyes open, right? There's this sort of drive towards sleep, but there's also a circadian component, which is why we sleep at night, whereas a mouse sleeps during the day. And so when those two things sort of coincide, the circadian drive, the sleep wake cycle, and the homeostatic drive, that's what initiates sleep. It's called the two process model. And so that's sort of how I got into it in grad school.
Yeah.
And then during my postdoc, I didn't do any sleep research.
Oh, wow.
I switched sort of fields to focus on glial cells, as you
Okay.
mentioned,
Yeah.
the non-neuron cells of the brain. And then when I started my lab back in 2020, we sort of merged to those two fields.
Okay. Yeah, so I was actually going to ask how you kind of like transitioned and merged those two fields, so I guess you naturally answered that question.
Yeah.
So my next question is a little more specific, and I try to do some digging to make it like you know, useful question. But I noticed that a study from your lab demonstrated the loss of BMAL1, I believe, if I'm saying it right, in oligodendroglia disrupts myelination and leads to fragmented sleep. And I wanted to ask you if you could walk us through how sleep and myelin integrity are interrelated at a cellular level and how we can connect this to long-term brain health.
Yeah. No, so yeah, that was a paper we published a couple of years ago where we were looking at interested in how the circadian clock. So we also, outside of this brain circadian clock, we have every cell in your body has its
Right.
own little molecular clock that oscillates on about a 24-hour cycle. And so we were curious how that clock would modulate these myelin forming glia. And since we know the circadian clock also influences sleep, we were also curious how it may then transfer into modulating sleep. At the time, and actually to date, there hadn't been anyone who actually had looked at the way these cells, these we call them oligodendroglia, so these myelin forming glia,
Right.
and the myelination that they produce influences sleep. And we were really interested in that question because myelination is this process of insulating the axons of neurons. So it allows what traditionally we think of as it allows for fast, efficient communication between neurons. So it's like insulating a telephone wire, allows the signal to actually propagate along the line. Same thing is true for myelination. And in disorders like multiple sclerosis and virtually every neurological disorder, there's changes in these myelin phenotypes which interrupt this efficient communication of neural signals. And why it's important, especially in humans, is because we have signals that need to converge from really distant parts of the brain in order to initiate a physiology. In this case, sleep and the sleep network that regulates sleep is really a brain-wide network. And so it made sense to us that this process of myelination would influence sleep and sleep architecture because of the nature of what it does, which is help signal convergence. And so that's where that study came from. And sure enough, what we found is when we disrupted BMAL1, the circadian clock in these cells, and we disrupted the ability of these cells to become myelinating oligodendrocytes and myelinate, we saw this very interesting sleep architecture change in the form of their inability to sort of maintain long bouts of wake, And that was driven by more like shorter, more frequent bouts of non-REM sleep, which
Okay.
is the deep regenerative sleep that we have.
Okay. So I was looking at some other podcasts. I noticed on Stanford's podcast From Our Neurons to Yours, you talked about extended napping and how in aging that could be an early indicator or even a contributor to neurodegeneration. So I wanted to ask you, what's your perspective on how napping or other like sleep alterations interplay with these neurodegenerative processes?
Yeah, it's a really interesting field at the moment is this intersection of sleep and neurodegenerative disorders,
Yeah.
especially Alzheimer's disease, because what we now know is that sleep alterations may be a symptom of sort of Alzheimer's disease that emerges long before the traditional symptoms that we think of with Alzheimer's. And so it's a pretty hot topic of study at the
Yeah.
moment, and we don't really know how or why it's playing a role, but there's a couple of different angles. One is we know that sleep has a lot of functions. One of them we believe might be to sort of clear out the waste of the brain potentially. If you've heard of glymphatic systems and things like that. But there's also this restorative nature to sleep that helps us to sort of rejuvenate and restore the brain after a long day of working really hard. We also know that sleep helps consolidate memories, which in Alzheimer's disease, memory dysregulation is and dysfunction is the prominent phenotype. And so we don't really understand mechanistically what cells are causing the sleep disruption early in the disorder, but we think the glial lineage may be playing a role in this
Okay.
sleep phenotype and propagating some of the traditional or typical AD pathophysiology that we associate with this disorder.
Okay. That's very interesting. So, based on your other findings in the lab, in the near future, do you foresee any interventions, whether that's behavioral, pharmacological, or chronotherapeutic, capable of preserving health or glial health, really, to be specific, or other like cognitive functions relating to sleep?
Yeah, I mean, that's the goal. That's the
Right.
hope.
Yeah. Yeah.
Um, I think there's a lot of interesting avenues at the moment. One of the things that we're really intrigued by with this intersection of myelin or oligodendroglia sleep is that it could be working through multiple different mechanisms. So, as I mentioned, these cells, these oligodendrocytes create myelin. But the cells that give rise to the myelinating oligodendrocyte are a precursor cell population called the oligodendrocyte precursor cell or OPC. And
Okay.
what's really interesting about OPCs and some of the things we're working on in the lab is that these OPCs can actually also form functional synapses with neurons. So most people, even most neuroscientists, think that neuron synapses only occur between two neurons, but they actually can also occur between neurons and OPCs.
Wow.
And we don't know what the function of these neuron OPC synapses are. We don't know
Yeah.
really how they integrate information within the brain. We don't really understand their function of them, but we're really curious about whether they could also be contributing to regulating sleep circuitry. And so there's multiple layers at which this lineage could be influencing this sort of neural circuitry that regulates sleep, from these neuron OPC synapses to myelination, but also these oligodendrocytes provide metabolic support to neurons. So it could be a metabolic issue as well. And so I think there's many different ways that this lineage could be influencing sleep
Yeah.
circuitry, and that's one of the things we're sort of probing. And depending on if any of those processes are actually regulating it, your
Right.
therapeutic approaches could be very different, but they could be very broad potentially in their influence.
Okay. So I know we've been talking about your lab a lot, so I want to backtrack a little bit. So I know in the lab that I worked at here in WashU, we use some cool stuff like Firefly, Luciferase, PMT machines, and all kinds of stuff. So I was just curious, what kind of technology or innovative methods do you guys use to help with your projects?
Yeah, we have a pretty broad range of techniques in our lab because we're interested in questions from molecular all the way up through behavioral and circuit function. So we do everything from really heavy molecular techniques like sequencing involving things like RNA sequencing, both single cell and bulk. We do metabolomics because we're interested in
Okay.
metabolism. We do lipidomics because myelin is a lipid-rich content. So understanding the building blocks of myelin, the lipid content of
Yeah.
these Myelin is important to us. We do a lot of genetic manipulation, whether it's mouse modeling, where we can go in and selectively knock out or induce loss of function of a specific gene in a specific cell type. In this case, it's usually an OPC or an oligo. So we can create those mouse models. We can also use genetic techniques in vitro, so we can isolate these cells and look at them just in culture, and we can disrupt genes using CRISPR or siRNAs. And then we can, also do in vivo work. So we can do these manipulations in mice. We can look at sleep in these mice using EEG, EMG biotelemetry. So lets us record sleep wake activity in the brain so that we can look at sleep wake cycles within these mice. We do electrophysiology so that we can actually look at these neuron OPC synapses and synaptic activity within these neuron OPC synapses. We do behavioral studies then to look at whether or not it changes their behavior. So if they're more anxious-like, or if they have memory deficits or motor deficits, because these disrupting myelin or oligodendrocytes could impact those things. And then we have a technique that's really unique to us because we're also very interested in how this relates to human biology.
Right.
So we actually can do stem cell work where we can actually take individuals who have various neurological disorders, and we've done this with autism spectrum disorder, 22q deletion syndrome, which is a disorder in which individuals are highly likely to develop schizophrenia, but are very likely to also have autism or epilepsy, multiple sclerosis, Alzheimer's disease, and we can take these inducible pluripotent stem cells from these individuals. So we take blood from them, make them into these inducible pluripotent stem cells, and then in our lab, we can differentiate them into human oligodendrocytes.
Wow.
And then we actually can take those human OPCs or oligodendrocytes from individuals with various brain disorders, and we can actually place them on a system in which we take a brain slice of a mouse called the shiverer mouse that lacks its own endogenous compact myelin because it has a mutation in a really important myelin gene called myelin basic protein. And within a few weeks, those human oligodendrocytes will start to myelinate the axons in that mouse brain slice. And so we can do these
Wow.
complex analyses of oligodendrocyte morphology and myelin architecture in terms of the length of the myelin, the complexity, the thickness, things like that. And so it's for the first time, it allows us to translate some of the things we may be seeing in mouse models into human diseased myelin models.
That's super cool. I'm just curious, how long has it been since you guys were able to do that?
We published this paper, let's see, a couple months ago.
Okay.
It's building off of previous work of other individuals, especially Anna Williams' lab. We published the paper with Anna Williams' lab at University of Edinburgh. So, but we have some really interesting findings for a number of disorders because as a field, it's the first time we can actually really sort of investigate how human oligodendrocytes in these disease contexts may vary in their myelin capacity.
Yeah. So, this is more of like a futuristic question, but what's next for the Gibson lab regarding sleep and glial biology? Are there any emerging questions or technologies that you haven't mentioned previously, I guess, that you're particularly excited about?
Yeah, one of the things we're really excited about, I think, is just really trying to dive into understanding how this lineage, oligodendroglial lineage, from the OPC all the way up through the myelinating oligo, regulates sleep. And so we have a bunch of different studies, like I said, looking at the role these synapses might play, especially related to calcium signaling and sleep, because we know that calcium signaling and a main driver of the arousal state called norepinephrine, which comes from a group of cells in this brain region called the locus coeruleus, influences OPC dynamics and how that may influence these neuron OPC synapses and potentially how that might contribute to sleep, all the way up to just how different models in which we disrupt either the production of oligodendrocytes or myelin capacity of those oligodendrocytes will influence sleep architecture. And so I think there's a lot we don't know. We don't know really anything minus the what we published a few years ago. And so there's a lot of room to really understand that.
Yeah.
And then I think how that changes in these various disease contexts. Not only a neurodegenerative disorder, which we know sleep and AD are very interconnected at the moment, but we also know in neurodevelopmental disorders like autism spectrum disorder, sleep disturbances is one of the leading sort of cause of concern for a lot of individuals. So these sort of sleep and myelin phenotypes are coexistent in virtually every neurological disorder. And so understanding how those two things intersect in normal biology and then maybe become disrupted in these neurological disorders is a real driving force of our lab.
Yeah. In the next 10 years, where do you see like sleep medicine as a whole going? Like, I guess beyond the lab. Um,
Yeah, I think sleep research and sleep medicine has traditionally, like most of neuroscience, been very neuron centric.
yeah.
And glia are sort of late to the game, not
Yeah.
for lack of interesting things, but relatively compared to neuron focused research,
Right.
glia are newer. And the majority of the cells in the brain are glia.
Yeah. 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 It's
And so
interesting.
I think we've been missing half the puzzle.
Right.
Um,
More than half, I guess.
more than half the
Getting
puzzle,
numbers. Yeah.
and neurons can't do what they are meant to do without glia
Right.
there to
Yeah.
help them. And so I think we're going to see, personally, I think we're going to see a big explosion in
Okay.
our understanding of not just sleep, but most neurological disorders. And
Yeah.
because of the incorporation of glia and the way they modulate neural circuitries and neurons themselves.
Yeah. I remember the first thing that I learned or like read in one of the textbooks when I first started studying neuroscience was the fact that glia were the vast majority of the cells in the brain. And I
Yeah.
was like, wow, I mean, I've never been, this was like in eighth grade. So for like the first like 14 years of my life, I just never even knew about this cell that was like very important in the brain. So I think it's
Yeah, and they're a class of cells. They do a
right.
whole host of things
Oh,
from
yeah, yeah, yeah.
modulating the synapses between
They have so
neurons
many functions.
to immunomodulation
Right.
and blood-brain barrier stability and myelination. So they're
Yeah.
they're they're clearly doing, they're not these passive glue-like
Oh,
cells. They're
no.
quite active. And I think because of that, incorporating them into our understanding of neural circuitry is really going to, I think, aid in our understanding of
Yeah.
neurological disorders for the next 10 years.
So, on that note, thank you, Dr. Gibson, for joining me today. I think we had a great conversation, and I'm really glad you were able to share your knowledge and your passion for your work on the podcast today. It was a pleasure
Yeah,
having you on.
thanks for having me.
Yeah.